Not here, but all around the room. Oh, it's coming. Okay. So yeah, I've been giving these lectures for a number of years, so I guess I've had exposure to antibodies during my research, and so it was quite logical that originally Dr. Lund asked me to do these. So let's get started. So we'll have a break at some point halfway through because we've got 2 hours, and I don't want to kill you all. Okay. So here, just by way of reminder, is the structure of an antibody. I'm getting some feedback here, aren't I? How's that? Okay. Yeah, here's the structure of an antibody as a reminder. So an antibody is made up of 2 heavy chains here, and they're disulfide linked together. And there are 2 light chains, and these are disulfide linked to the heavy chains. In red, we have these so-called variable regions. These have the antigen binding regions as part of them. And then we have the FC region here. And each of these blobs is an immunoglobulin superfamily domain that makes up these antibodies. Okay, and in a space-filling model of the structure, here we have the 2 heavy chains shown in this purple colour. Actually, I think running through like that, and then here's the other heavy chain, and the light chains are shown in this kind of pinky colour. And these IgG1 antibodies have an N-linked glycosylation site here in yellow, and we'll get onto the glycosylation in the lectures next week and how that can be important. Okay, so what you need to know from these lectures then. So I will tell you about the strengths and limitations of using antibodies as therapeutics, I guess, as opposed to small molecule inhibitors. And then I will give you 3 examples of antibodies that have really led the way and are being used in the clinic. And I'm going to focus on cancer. Okay, so this is what I'll tell you. So I'll introduce you to therapeutic antibodies, possibly going over some of what you've already learned. And then these 3 blockbuster antibodies that are in the clinic to treat cancer, I'll go through the story of how those antibodies came about. So Avastin, Herceptin, and Rituxan. Okay, so as you have probably learned already, therapeutic antibodies are the fastest growing class of therapeutics, so they will outstrip small molecule inhibitors. They may already have outstripped in terms of sales. And we've got marketed antibodies that have been approved to treat cancer, autoimmune diseases, inflammatory diseases, and also rare medical conditions. And there's a great variety of different structures that are now emerging, and I will touch on that in the lectures next time. But there's a variety of ways that antibodies can be modified to make them even more useful. And the big 5 blockbusters that have kind of led the way and made so much money for the companies that made them are indicated here. So the ones in red are the ones I'm going to focus on. They just happen to target different cancers. But I'll just briefly mention these 2, so Remicade and Humira. These are both antibodies that target tumour necrosis factor. So tumour necrosis factor is very important in inflammation, in the immune response, in driving inflammatory diseases like rheumatoid arthritis, where we get attack of our joints, Crohn's disease, so this is an inflammatory bowel disease, and plaque psoriasis. This is where it's inflammatory disease of the skin, and you get these nasty scaly patches. But antibodies to TNF-alpha have been shown to mop up the TNF, reduce the inflammation, and have been very important for treating these inflammatory diseases. The other 3, Herceptin, Avastin, and Rituxan, are all targets for cancer. So, of course, cancer is one of the big diseases, the big killers of people. A very famous one, Herceptin, is used to treat breast cancers by targeting epidermal growth factor receptor 2, also rather confusingly called HER2. We have Avastin. This targets vascular endothelial growth factor. This used for a variety of different solid tumours. And then we have Rituxan. This targets a cell surface protein called CD20, and it's being used to treat rheumatoid arthritis and also a type of blood cancer called non-Hodgkin's lymphoma. Getting this strange kind of booming, strange sounds, like whale sound in the background, isn't it, when I talk? You enjoying the whale sound? It's quite good, isn't it? I'll just carry on. This is turning into the best lecture I've ever given, I think, because of the whale sound. Okay, so just a bit of nomenclature. So we've got our OMABs, our XIMABs, our ZUMABs, and our MUMABs. So an OMAB is purely a mouse antibody. So this will have been raised in mouse against a human antigen. Then we have chimeric antibodies, which are, they have the mouse antigen binding region, but the rest of the antibody is human. Then we have humanized, so this is just where we have it's effectively a human antibody, but just in these yellow stripes here, we have the complementarity determining regions, the antigen binding regions from the mouse sequence. And then finally we have perfectly human antibodies, so these could have come out of a phage display type of method of making antibodies. Have you guys come across phage display yet? Have you, as for making antibodies? I might talk about that next week. I was thinking of revamping my lectures for next week, so I might talk about a bit of phage display. But it's a method frequently used in pharmaceutical industry. They have big libraries of human antibodies that can be expressed by viruses, for example. So big viral libraries, and they can take a target and they can, by a screening method, they can figure out which of their millions of human antibodies are able to bind their particular target. And this is a way of generating human antibodies and avoids the need for having to go through a mouse and making hybridomas in the classical way. Okay, so the mouse antibodies, they're of course 0% human as opposed to the human antibodies, 100% human. And the chimeric and human have varying degrees of percentage human sequence. You have to give the mouse antibodies quite frequently because you'll get an immune response made against them in the human, and so you have to keep giving them, whereas a fully human antibody, it'll last a long time in the blood of the patient, and in theory you don't have to deliver that as often. And similarly, repeated administration is difficult for mouse because you're just continually getting this immune response. But in theory, if you wanted to, you could give the human antibodies very often and you wouldn't raise an antibody response against those antibodies. And in terms of effector function, so this is CDC is complement-dependent cytotoxicity, and I'll come to that later on. And ADCC is antibody-dependent cellular cytotoxicity. Again, I'll come to that. Human antibodies are very good at eliciting that because it's the FC regions of the antibodies that recruit immune cells to do this sort of cytotoxic action. But mouse antibodies aren't very good at doing that because they have the mouse FC portion that doesn't bind very well to the human FC receptors on the immune cells. And I'll come on to that later. And just maybe an even clearer image, this is from a company called Absolute Antibody that I've dealt with. So the OMAB antibodies, purely mouse. Any antibody with XIMAB on the end will be chimeric, so it'll have the mouse IG domains that bind antigen, but the rest is human. The humanized just have the complementarity determining regions from the mouse that bind antigen, the rest is human, and finally a fully human. And these are made by genetic engineering, and Absolute Antibody happens to be a company near Oxford that will do a lot of this for you, so you can give them an antibody that you've made in mouse, and they will humanize it for you at a cost, of course. They will also sequence your antibodies for you and give you your sequence. So this is one thing we've done in the lab. We've made monoclonal antibodies in mouse. And, you know, the thing you do now, if you make a monoclonal antibody, you get it sequenced. So we've got the cDNA sequence. So in theory, we can make that recombinantly, or we can start doing engineering on that antibody. And it's a nice, feels safe now. You know, we've got the sequence of our precious antibodies. So even if there was some sort of liquid nitrogen freezer disaster where we lost all our hybridoma cell lines that are producing this antibody, at least we've got the sequence and the antibody isn't lost, and we could make an expression construct for that antibody and start producing it recombinantly in a cell line system. Okay, if you're thinking about embarking on a project to make a therapeutic antibody, how do you select what target to make an antibody to? Well, there are, I guess, 3 broad things you could target. You could go for something that's already clinically validated, so already antibodies out there to the target. You could go for experimentally validated targets, so this is where you're pretty sure it's going to work, but they haven't been targeted yet. And finally, the sort of thing that we do in the lab with basic research, we try and find new functionally validated targets. And I'll go through each of these in turn. So yeah, this is when you plan talks, you just often get a bit bored and try and find funny images. So here, this is my image of the clinic. So we've got a doctor and a funny-looking baby here who looks rather intelligent and all-knowing. Anyway, the idea of taking clinically validated targets is you want to beat the blockbusters. So those 5 big blockbusters I told you about, well, you could try and make an even better antibody that targets TNF, for example. So develop 2nd or 3rd generation antibodies that work better than those that are already in the clinic. So this has low risk because you're almost certainly going to be able to make antibodies that do target what you want, and they'll probably have some sort of therapeutic effect. But there's lots of competition. You know, you've got to somehow make an antibody that works better than the existing ones, which can be pretty difficult. So the second thing you can do is you can go for experimentally validated targets. This is my little caption of an experiment. This is what will happen in your final year projects, or you MSc students, when you finally get to the lab. And I guess the MSci students, this is already happening to you all the time, isn't it, this sort of thing? Yeah, they're all nodding, those of you up here, yeah. So typically you might take targets that are a really good bet. So for example, cytokines or associated receptors that are well known to drive disease processes. So if you were to make an antibody against them, there'd be a great chance of you being able to treat that particular disease. We really need new targets in cancer because cancers are quite complex and they frequently develop resistance, and it's becoming clear that maybe in the long term, if we have multiple therapies that we can hit the cancer cell with at the same time, that might give us the best chance of success. And finally, there's functionally validated targets. So this is maybe the sort of thing you'll be doing in your lab projects. So maybe doing some sort of functional screen to try and find antibodies that could have an inhibitory effect. For example, you could make antibodies against a tumour cell line and screen for antibodies that inhibit growth of that tumour. You could then try and figure out what protein that antibody is recognising, and then ask the question, what sort of pathway are you hitting, and could this be a new way of targeting the tumour? So it has great potential, this sort of approach, but it's very high risk because frequently you might end up with nothing, and even if you do get something, what are the chances that that will ever really get through all the stages of clinical trials and ultimately become a drug? It's kind of very low chance, but it's interesting, I think, to do this sort of research, and at the very least you might get a top quality publication. So you might find a new way of inhibiting a cancer cell, and at the very least get it published in a good journal, even if it doesn't eventually make it all the way through to the clinic. Okay, so here's just a timeline, just to give you an idea of what was happening before you were all born in this sort of field. So back in 1975, Milstein and Kohler in Cambridge were able to figure out how to make monoclonal antibodies in mice using their hybridoma technology. And then they got the Nobel Prize for that in 1984. And the first antibody chimerization, human and mouse, was described back in 1984. So other notable developments. So we had the first mouse antibody with therapeutic action back in 1986. This was against CD3, that's one of the chains of the T-cell receptor. So here they were targeting immune cells, probably in some sort of anti-inflammatory type of approach. We had antibody humanization described in 1986 as well. Phage display technology came about in 1990. So this is where I mentioned you have big libraries of human antibodies and you screen them to find the antibody you want, so you don't have to go via the mouse hybridoma approach. So another way to make humanized antibodies is with transgenic mice that have the machinery to make human antibodies. So this was described in 1994. And then various therapeutic antibodies started to come online, late 1990s and the 2000s. So the first therapeutic antibody from a phage display screen came about in 2002. And then, so this only goes up to 2010, but you can see there's this steady stream of therapeutic antibodies now being generated, and we're starting to get second generation ones now that have modifications that make them slightly better than the original versions. So I won't go through all the rest of this detail. But this table just gives you an idea of the number of new antibodies that are coming out. So from 2011 to 2016, you can see we've got 3, 4, 5 each year new therapeutic antibodies coming onto the market from these various companies. And you can see the various UMAB, CMAB type of nomenclature to see exactly what type of antibody they are. Okay. So the advantages of therapeutic antibodies over, for example, small molecule inhibitors is that we've now got great established methods for making these antibodies, either in mice or using this phage display technology. And we're really learning how we can now get the sequence of our antibodies, the DNA sequence, and how we can modify the antibodies to give them new properties. They have a higher success rate compared to other drugs, I guess because they're natural proteins. Our blood is full of natural antibodies, so they're well tolerated. And we've got very good established production technologies for producing large amounts of the antibodies. And we've got good experience, and our knowledge is ever expanding to allow us to generate future therapeutic antibodies. There are some limitations, so they're expensive, so small molecule inhibitors, very cheap to produce, but antibodies do have high production costs, and commonly you need large doses, and this can limit patient access and clinical applications. It can just be too expensive to treat a large number of patients. You're currently limited to cell surface or extracellular targets because you can't get antibodies across the plasma membrane inside the cell. And you can't just take them like a pill, like you would a small molecule, because if you, once they get into the digestive system, they'll just be digested. So they have to be injected into the bloodstream. And they're big, and this large size can really limit the extent to which they get into certain tissues where they might be needed. And then we've got the problem of the blood-brain barrier. So if we're thinking of trying to treat diseases like Alzheimer's, how do we get our antibody across the very tight blood-brain barrier? So that's a problem. So there are some definite limitations. Okay, so now I'll go on to the second part then, and I'll go through each of these success stories, these blockbuster antibodies, Avastin, Herceptin, and Rituxan. Okay, yes, so here's our Avastin first. So this is a target for many solid tumours, and the antibody targets vascular endothelial growth factor A. So I'll go through that now, how that was developed and how it works. So here's the rationale behind Avastin. So I might try and just turn the lights down a fraction. Is that okay? That's too dark, do you think? There should be a way of making it a little bit... Oh, hello. There's a very complicated scheme of lights here that I will attempt. Is that better? Not too sleepy? Okay. Right, so here we have a solid tumour, and once, so these solid tumours will develop out of our epithelial cells in the body, and once they get to about 2 or 3 millimetres in diameter, if that tumour is going to go on and proliferate and become bigger, it has to now attract its own blood supply. So once it gets to a certain size, it needs a blood supply in order to survive. And the way it does that is by releasing vascular endothelial growth factor. And that induces a process called angiogenesis. And angiogenesis is the growth of new blood vessels from existing vessels. So here there may have been a vessel going quite close to the tumour. When the tumour releases this VEGF, which these little dots, this will cause sprouting of new blood vessels, which will envelop the cancer and nourish it with blood, oxygen, and nutrients, and allow it to grow on. And also, importantly, give it a means to metastasize and get to other parts of the body. Because as you may know, invariably, it's not the primary tumour that kills you, it's the metastases that have spread from that primary tumour. So the idea of Avastin is it's an antibody developed to target VEGF, this cytokine that, this growth factor that causes angiogenesis. So the idea is we're going to mop up all the VEGF, we're going to stop this process of angiogenesis, and we will starve the tumour to death. And this still is really exciting. It's a great idea. In theory, you could kill off all solid tumours in this way, regardless of where they happen to crop up in the body. I guess the brain could be a problem with the blood-brain barrier, but in most places you might think this might work. So just a little bit more background on angiogenesis. So here we have our blood vessels. So we've got our green endothelial cells that line all our blood vessels. And then the way angiogenesis starts is we get a tip cell that has detected VEGF, this growth factor. It starts to sprout, and then it will form a tube as it migrates and starts to proliferate and move outwards, and that's how we get our new vessel developing. And a key transcription factor in all this is HIF-1 alpha. So in a normal state, if we've got cells in our tissues that are quite happy, they've got a nice, relatively high oxygen environment, this will cause them to have low levels of the transcription factor HIF-1 alpha, and they won't be producing any VEGF. But as soon as they go into a low oxygen state, their normal response is to upregulate this transcription factor called HIF-1 alpha. One of the things that HIF-1 alpha turns on is transcription of VEGF. So they start releasing this growth factor VEGF, and that causes angiogenesis to get blood vessels growing nearby and to replenish their oxygen supply. So this is a very natural response in the body to a variety of insults, but it's something that tumour cells have hijacked as a means of attracting their own vasculature as they reach a certain size. So in adults, it's, this angiogenesis is quite normal, so in the reproductive cycle, and also when we injure ourselves, the whole process of wound healing, we need to regrow that vascular system, so it's a VEGF driven process. Also, I have an exciting movie now to show you. At least I think it's exciting. I've forgotten what it shows from last year.
The body must generate. As a normal part of growth and development, the body must generate new blood vessels to oxygenate the tissues. In a process called angiogenesis, new vessels sprout from existing ones. In this movie, we see endothelial cells sprouting to form new branches from the aorta of a zebrafish embryo. Each sprout is initially formed by one or a few endothelial cells. The process begins when an endothelial cell of a small vessel is activated by an angiogenic stimulus, such as vascular endothelial growth factor, or VEGF. In response to the stimulus, the endothelial cell becomes motile and extends filopodia that guide the development of a capillary sprout. The leading or tip cell continues to move away from the capillary as cells behind it migrate in and divide, forming a stalk. The sprout begins to hollow out, forming a tube. In this process, pinocytic vesicles fuse with one another. The large vacuoles formed in this way then fuse with one another, creating a lumen that runs through the capillary sprout. In culture, endothelial cells behave in a similar way. They spontaneously develop internal vacuoles that join up from cell to cell, creating a single lumen shared by many cells. In the example shown here, the individual cells contain either a red or a green fluorophore. Note that the areas of green and red are distinct. Even though cells share a lumen, they do not share cytoplasm and remain separate cells after the fusion events. Angiogenesis is critical not only in normal development and wound healing, but also in the development of tumors. A tumor must stimulate blood vessel formation to grow more than a few millimeters in size. VEGF is a key activator of angiogenesis in both normal cells and tumors. When cells within a tumor become oxygen deficient, they begin to express VEGF. VEGF diffuses through the tissues, activating endothelial cells on nearby vessels. This results in capillary sprouting. Some new cancer therapies are targeted to block the action of VEGF, with varying clinical results.
Oh, serious music at the end. I've forgotten that. Okay. So, hopefully that's all clear on angiogenesis. So a bit more background. So VEGF is a growth factor, and so it will bind to receptor tyrosine kinases on the endothelial cells and trigger them to proliferate and undergo this angiogenesis process. And there are different types of VEGF. So here you can see the two prominent ones, VEGF-C and VEGF-D. They bind VEGF receptor 2, that's the major one on blood vessels. Blood vessels also have VEGF receptor 3, but on lymphatic vessels, the main one is VEGF receptor 3. And lymphatic vessels are equally as important because the tumour needs a means to get rid of its fluid via the lymphatics, and also the lymphatics are a major route by which the tumour cells can spread by metastasis. They can travel out along the lymphatics, get to lymph nodes, and then use that to spread throughout the body. So this is what we aim to do with our Avastin, our antibody that targets the VEGF. So our Avastin, we hope, will mop up all this VEGF that's being released by the tumour It will kill off the blood vessels or prevent them forming in the first place. So we lose both the blood supplying vessels and the lymphatic vessels, and the tumour cell is going to shrivel up and die. That's the master plan with this approach. Oh, and so just a bit more introduction. I've forgotten I had all this introduction. So this is the way VEGF actually signals. So here we have our VEGF receptor. It's actually a receptor tyrosine kinase. So it has a tyrosine kinase domain intracellularly. When VEGF is dimeric, and when it binds to this receptor tyrosine kinase, it causes dimerization. We get transphosphorylation of tyrosine residues in the cytoplasmic tail of the kinase by the kinase domain, and this leads to a whole load of signaling pathways that I'm not going to get into, the RAS-MAP kinase pathway, PI3 kinase, and phospholipase C-gamma. And all this culminates in various changes in gene expression, so various transcription factors are activated. We get proliferation of the cells, increased survival, increased migration, remodeling of the cytoskeleton, all things you need to do to get this process of angiogenesis going. Okay, so how did VEGF therapy, how did Avastin arise? It's quite an interesting story, so I thought I'll go through it. So back in the 1980s, several groups actually discovered VEGF and showed that this was the critical growth factor for inducing angiogenesis, this growth of new vessels from existing vasculature. And then in 1993, the Ferrara group showed that they could make anti-VEGF antibodies, and that would actually block tumour growth in mouse xenografts. And then in 1997, we had our first humanised antibodies produced, and this one called bevacizumab is actually the Avastin. So the way the xenografts work is you take a mouse. This has to be an immunodeficient mouse, typically a nude mouse, so it lacks an immune response. And this poor mouse gets injected with a human tumour cell line, so it will now develop a tumour, and you can measure the size of this tumour growing just underneath the animal's skin. And the experiment that was done was to treat these mice that have received different types of tumour. So here we've got 3 different types of human tumour. And the minus and the plus refer to whether these animals have been given the anti-VEGF antibody, the plus, or just an irrelevant control antibody, which is the minus. And there must have been party time in this lab when they got this result, because it's really a phenomenal result, this dramatic reduction in tumour weight when they give this anti-VEGF antibody. So it seemed to be looking like the holy grail, maybe, of treating solid tumours. Could this be the way we're going to wipe out solid tumours by starving them of their blood supply with this anti-VEGF therapy? When they started to do experiments in human, they started to get slightly different results from the mouse. So this is a study where they're looking at growth of the tumour on the y-axis, and then just the time after the treatment. And what they're doing in this little preclinical trial, they're either giving the patients nothing, or they're giving bevacizumab, this is our anti-VEGF antibody, or they're giving capacitabine, I can never say that. That's a standard chemotherapy. It's a very nasty, toxic drug that will inhibit DNA synthesis and have lots of nasty side effects, but often that's the best thing you've got to treat this sort of cancer. So trying with various combinations, either the antibody on its own, the nasty cytotoxic drug on its own, or the various combinations of the antibody and the drug. So the poor controls, their tumours are developing. The anti-VEGF antibody has a little bit of an effect, but not very much, certainly nowhere near as good as the effect that they were seeing in mouse models. The nasty cytotoxic drug helps a bit, but what was really striking is when they combined the cytotoxic drug with the anti-VEGF antibody, they saw a rather nice inhibition of tumour growth. Still not perfect, the tumour's still growing, but what you find with a lot of these cancer drugs, they rarely wipe out the tumour completely, and it's just a case of slowing down the tumour growth and extending the length and quality of life in the patient. So this was still looking very good. So it entered the clinic, Avastin. So this is the anti-VEGF back in 2004. So this was the humanized anti-VEGF antibody. And it was always used in combination with one of these nasty chemotherapies because of this preclinical work showing that you couldn't use it on its own. It had to be with one of these chemotherapy agents to get a good result. So this 5-fluorouracil, this will be another one of these DNA synthesis inhibitors. And New England Journal of Medicine, you know, this is the premier journal for publishing clinical findings that really have a massive impact on work done in the clinic. So this was a phase 3 trial showing that you had a 4.7-month increase in overall survival. And that may not seem much, but actually in the business of cancer, this is seen as a great success to, on average, extend the patient's life by 4.7 months. So it was approved in 2004 as a first-line treatment for metastatic colorectal cancer. And then in 2006, it was approved for second-line treatment of colorectal cancer, again in combination with a chemotherapeutic. And there's loads and loads of trials currently going on for lots of different cell types to see how effective this Avastin treatment in combination with chemotherapy can be. And of course, this made big news. So here's an excerpt from the BBC News website back in 2004. So the US approves new cancer drugs. So it halts the growth of tumours by cutting off their blood supply. And it's been shown to prolong the lives of those with bowel cancer. Works by starving the cancer cells of blood, thus stopping them from growing. So unlike chemotherapy, which works by killing all cells, this drug only targets the cancer cells. So yeah, a very significant breakthrough in the fight against cancer. So it prevents the formation of new vessels. Actually, this is completely wrong, okay? So I'll tell you why it's wrong in a minute, but the side effects are pretty minimal. So Avastin on its own, side effects are quite rare compared to the nasty chemotherapy. And the side effects that we do get from Avastin are likely to be just because of its normal role in maintaining a healthy vasculature in the body. So patients tend to get high blood pressure, increased risk of bleeding, delayed wound healing, heart dysfunction, thrombosis, which is the aberrant formation of blood clots, and vessel regression in some organs. I guess all what you might expect if you're treating this important growth factor VEGF. And this is how it really works. So it's not cutting off the blood supply to the tumours, it's doing the exact opposite. So, and this is different to the mouse. So the original mouse work did seem to be showing that it was cutting off the blood supply to mouse tumours, but in human it works by the opposite mechanism. It actually promotes blood supply to the human. So even though it's doing the exact opposite of what was hoped, it still works, but only in combination with the chemotherapy. So here's our normal tumour, and this is the sort of blood supply it will have. It's this horrible, tortuous, often blind-ending vessels, going nowhere, slow blood flow, so hypoxia, so low oxygen, poor perfusion, high pressure. The tumour's just about surviving in this environment and growing, just about getting enough oxygen, but it's incredibly difficult to get chemotherapeutic agents into the tumour in this sort of environment. You're just not going to get good concentrations of the chemotherapy into your tumour. Instead, you're going to be targeting all other parts of the body with this nasty cytotoxic drug, and that's why you get such bad side effects. What the anti-VEGF, the Avastin antibody, does in human, it actually reduces the level of VEGF and it allows the blood supply to the tumour to be normalised. So it basically corrects this horrible, tortuous set of vessels into a much nicer set of vessels that are perfusing the tumour now very nicely with blood. And you might think, well, that's going to be a catastrophe because now we're suddenly feeding our tumour with lots of blood. But that's why the combination with chemotherapy is so important, because if we give these chemotherapeutic agents at the same time, the VEGF blockade normalises the vasculature, it allows blood to get properly to the tumour again, and that allows the chemotherapy drugs to get to the tumour and exert this cytotoxic effect. Yeah.
Why does it still cause benefit on its own then? Because it did cause slight benefit.
I knew you were going to say that, and I've never had that question before. So yeah, you're referring back to you're referring back to this, and you know my answer's going to be I don't know. I actually looked this up before. I was just flicking through the lecture before I came, and I thought, you know, I wanted a bit more information on what they were doing here. At first, I thought it might have been in mice, but I'm pretty sure it's human. And I couldn't find it. I could not find this Shen et al. In this. So I need to do a bit more digging. But maybe that's good outside reading for you. You know, if anyone can figure out exactly what. I mean, it may be that it's not significant. So you're talking about on its own having some effect. Again, I need to look at the paper to see if the stats they did made it significant. But it could be just a spurious thing. Yeah.
Another thing is treating with the chemotherapy first before the antibody also seems to have a better effect.
It does, doesn't it? So this is the chemotherapy and then the antibody. Yeah, and so the chemotherapy is going in here. Maybe it hangs around a bit, and maybe the effect of the antibody is quite rapid. It's a good point. Again, I need to go back to this paper. It's ironic that I've never had questions about this, and this was the year I thought, oh, I'm going to just get my mind clear on this, and I couldn't find the damn thing. But good questions. I like it, yeah. I'm sorry I can't give you perfect answers. Okay, so just to summarise the Avastin, so this cunning plan that they had to starve the tumour of its blood supply by mopping up this key angiogenic growth factor VEGF worked fantastically well in mice. In humans, though, it didn't work on its own. It did work in combination with these traditional chemotherapeutics. And now the widely believed thinking is that actually it's not destroying the tumour's blood supply. It's instead normalising it, giving the tumour a better blood supply, so allowing access for these chemotherapeutic agents. All right, so I think now we will have a quick break. So I'm going to pause. Shall we push on then, everyone? Everyone happy with Avastin? No more questions on that? So we will move to Herceptin. So this is an antibody that targets the receptor tyrosine kinase called HER2, also called human epidermal growth factor receptor 2, to treat breast cancer. So just a bit more intro on these receptor tyrosine kinases. So HER2 or EGF receptor 2, just like VEGF receptor, it's one of these receptor tyrosine kinases. So they're quite a large family. They are characterized by all having this intracellular tyrosine kinase domain. They've got a transmembrane region, and then they've got varying extracellular regions which can be immunoglobulin-like domains. So these are the same domain structures that make up antibody molecules, but they're frequently used to make up membrane proteins, and then other types of domain, such as these cysteine-rich domains for EGF receptors. So receptor tyrosine kinases are really important. Their signaling and conservation go all the way back to very simple organisms like worms. They're critical for cell proliferation, differentiation, survival, metabolism, migration, and cytoskeletal changes that cause changes in cell shape. And there are a number of mutations in the varying receptor tyrosine kinases that give rise to various important diseases, cancer, diabetes, inflammation, you name it. So a very important class of proteins in health and in disease. And in terms of cancer, this is how different receptor tyrosine kinases can give rise to cancer. So we can get amplification of expression of a particular receptor tyrosine kinase. So this is where we express, or a cell expresses much higher levels than it should be. And this can lead to increased signaling and driving the cell into a highly proliferative cancer-type state. Mutations can arise in these receptor tyrosine kinases, which cause ligand-independent firing of the kinase. So these can either be point mutations, as indicated by this little red dot, or truncations where you lose most of the extracellular region, and now this receptor tyrosine kinase is able to be constitutively active in the absence of any ligand and can drive a cell to uncontrolled proliferation and cancer. And then finally, we can have a scenario where a cell aberrantly starts to produce a growth factor for a receptor that it actually expresses. So in a normal case, cells have growth factor receptors, so these receptor tyrosine kinases, and they may also produce growth factors, but they don't produce the growth factor for one of their own growth factor receptors, because that would cause uncontrolled growth. But if mutations do cause them to produce a growth factor for a receptor tyrosine kinase they have, this can cause this autocrine loop of constitutive activation and driving cell proliferation and driving cancer. So this is a table just taken from, I think, the Weinberg cancer textbook, just showing you some of the important receptors which can drive cancer when they're mutated. So you'll see that EGF receptors, also called ERB or HER receptors, are very prominent here. So there are 4 of them. So number 1 in the family can be overexpressed to drive a variety of different cancers. Number 1 in the family can also have its ectodomain truncated by a mutation to drive other types of cancer. Number 2, which we're going to be talking about, called HER2 or ERB2, overexpression is responsible for a phenomenally high number of breast cancers, so 30% of these breast cancers. And numbers 3 and 4 in the family also can drive a cancer when overexpressed. And there are examples of other receptor tyrosine kinases, like the FGF receptor, driving others. But it was really realised back in the late 80s that HER2 or ERB2 overexpression was really linked with poor prognosis in breast cancer. It was really bad news if you presented with a breast cancer that had ERB2 overexpression.